6 people squeezed through a gap in a cave wall in western Romania in the autumn of 2023 and came out 6 hours later having photographed something that 31 independent researchers across 9 countries have since spent almost 2 years trying to explain. Not explain away, actually explain. And not one of them has succeeded. What those 6 people found inside a sealed limestone chamber in the Tartaria karst system has produced 4 separate findings, any single one of which would be significant on its own. Together, they are forcing a fundamental rethink of what humans were doing, who humans were living alongside, and what humans were capable of 40,000 years ago. And the technology that has been brought to bear on this cave since that day of discovery is producing answers that raise more uncomfortable questions than the cave itself did. The chamber had been sealed for 40,000 years. The calcite formation that closed it off from the rest of the cave system had been growing slowly across the passage opening since before agriculture existed, before pottery existed, before any city anywhere on Earth had been built. Everything we call human civilization, every empire, every language, every religion, every book, every invention, every war, every discovery, happened after that passage closed, and something was left inside before it did. This is the story of what was in there, what the technology found when it analyzed it, and why the scientific community that has spent almost 2 years reviewing the evidence is still not comfortable saying out loud what the data appears to be saying. Let us start with where this is and why nobody was expecting it. The Tartaria karst system sits in western Romania, a region that carries an unusual archaeological reputation already. 3 kilometers from the site where this 2023 cave team was working, a Romanian archaeologist named Nicolae Vlasa discovered 3 small clay objects in 1961 in a Neolithic context dating to approximately 5,500 years before the Common Era. These objects, known as the Tartaria tablets, bear incised symbols that some researchers have argued represent the earliest known writing in the world, potentially predating Sumerian cuneiform by over 1,000 years. That claim remains contested. The debates around it have been running for 60 years. The point is that the Tartaria region had already established a pattern of producing findings whose implications the academic mainstream was not entirely comfortable receiving. Eastern Europe generally, and Romania specifically, has not historically been the place where the field expected to find evidence of sophisticated Paleolithic symbolic behavior. Western Europe dominated, France, Spain, the famous caves of the Franco-Cantabrian tradition, Altamira in northern Spain with its painted bison, Lascaux in the Dordogne with its hundreds of animals covering the walls and ceilings, and above all, Chauvet Cave in southern France, whose extraordinary paintings of lions, bears, rhinoceroses, and horses have been dated to somewhere between 33,000 and 37,000 years ago, making them among the oldest confirmed examples of sophisticated representational art ever found anywhere on Earth. The dominant picture, built from over a century of research centered on Western Europe, was that the most remarkable expressions of early human cognitive capability emerged in France and Spain and spread from there. Romania was not in that picture. Romania was exactly where the picture got complicated. The team that entered the Tartaria karst system in the autumn of 2023 was conducting routine geological and archaeological survey work. Their mandate was to document the stratigraphy, collect samples for dating, map the passages, and write a report. The kind of careful, systematic work that fills gaps in regional site inventories, without usually producing anything that ends up being discussed at international conferences in 9 countries. They were not looking for anything extraordinary. Nobody was. On the 3rd day of the survey, they found a draft of air coming through a gap in a calcite formation that had grown across a passage opening at some point in the deep past, a sealed chamber beyond it. The calcite barrier was not completely intact. A section had collapsed, leaving a gap large enough for a person to squeeze through, but positioned awkwardly enough that recreational cavers who had used the main cave entrance had apparently passed the area for years without investigating it. The survey team noticed the air movement, understood what it indicated, and made the decision to enter. The team later described coming into the chamber as a professional experience that takes a significant amount of time to process, because what you are seeing does not connect to any category of thing you have trained yourself to expect. The chamber was roughly 8 meters wide and 5 meters high at its peak. Not enormous, but substantial enough that a group of people could have occupied it comfortably. The floor was undisturbed. The walls were covered in paintings and engravings that had been sitting in total darkness, in sealed, stable conditions, at constant temperature and humidity, with no light, no air movement, and no human contact for 40,000 years. The initial photographic documentation that first day was limited by what the team was carrying for a routine survey. Mobile phone cameras, 1 DSLR body carried by the team's geologist, handheld LED lighting designed for passage mapping, not detailed surface recording. The images from that session were consequently imperfect in ways that complicated the subsequent analysis, but they were sufficient to establish the basic facts of what the chamber contained. And those basic facts were enough to generate the phone calls that brought specialist teams to the site within the following 3 weeks. The chamber floor preserved what appeared to be a structured deposit of materials, arranged in a roughly circular pattern around a central area that showed discoloration consistent with repeated burning over an extended period. Not a single fire. A surface used repeatedly enough to stain the underlying sediment black to a depth the initial probe measurement suggested extended at least 20 centimeters below the current floor level. This was a place people came back to again and again over a span of time significant enough to produce that depth of staining. Around this central burned area, the deposit included worked bone fragments, pieces of deliberately shaped ochre in red and yellow varieties, 2 large flat limestone slabs bearing incised marks that the team's archaeologist recognized immediately as intentional rather than geological, and a quantity of dried plant material that was subsequently sampled for analysis and produced radiocarbon dates that remain at the polite center of considerable scientific controversy. The ceiling and upper walls were covered in a combination of painted and engraved imagery. The full photogrammetric documentation survey, conducted by the specialist team over 8 days, cataloged 237 distinct graphic units on the chamber surfaces. 237, ranging from simple lines and geometric forms to complex multi-element compositions that the team's comparative analysis found no direct parallel for in the entire European Paleolithic cave art corpus. That finding alone would have been significant. It was not the most significant thing in the chamber. Now let us talk about dating, because the dates are where this becomes genuinely framework-shattering, and it is important to understand exactly how confident the dating programs were. Cave art dating is a methodologically complicated enterprise, and anyone presenting dates with unqualified confidence has not read the methodological literature carefully enough. That caveat established, the Tartaria chamber has been subjected to 4 independent dating programs using 3 different methodological approaches. And the results from all 4 programs are consistent with each other in ways that leave the researchers involved with very limited room to retreat into methodological uncertainty. The most significant dating method applied was uranium-thorium analysis of calcite flowstone deposits that have grown over portions of the painted and engraved surfaces. Flowstone is a mineral formation that builds up slowly in cave environments as calcium-rich water flows across surfaces. It grows over things that are already there. When you uranium-thorium date the flowstone that has grown directly over a painted surface, you are establishing a minimum age for the painting underneath. The painting has to be at least as old as the flowstone covering it. The flowstone dates from the Tartaria chamber cluster consistently in the range of 38,000 to 42,000 years before present. The imagery beneath those flowstone deposits is older than the flowstone. It is at minimum 38,000 years old. 38,000 years. That number places the Tartaria chamber art at the very beginning of the Upper Paleolithic in Europe. It is contemporary with, or potentially older than the earliest dated images at Chauvet Cave in France, which has been studied intensively for 30 years, and which currently holds the title of the oldest, well-documented painted cave tradition in the established research literature. Contemporaneity with Chauvet would already be remarkable enough by itself. What makes the Tartaria chamber something different and considerably more disorienting is the character of the imagery itself. Chauvet Cave is extraordinary. The paintings there are sophisticated, detailed, and compositionally complex in ways that continue to surprise researchers 3 decades after the cave's discovery. But they are recognizably part of a tradition, animals depicted with attention to anatomical accuracy. Horses, lions, bears, rhinoceroses rendered in ochre and charcoal with techniques demonstrating considerable skill and a clear representational intention. The Franco-Cantabrian cave art tradition has its own internal logic, its own conventions, its own stylistic vocabulary that appears consistently across hundreds of sites spanning tens of thousands of years. The Tartaria chamber imagery does not fit that tradition. The specialist team ran the 237 documented graphic units against a reference database of over 3,000 documented Paleolithic graphic units from sites across Europe and Asia. The representational elements in the chamber include animals rendered in a style that is immediately recognizable as intentional and skilled, but that carries proportional distortions and compositional relationships between figures that do not appear in comparative material from any contemporaneous or earlier European site. The abstract geometric elements are more troubling. A set of recurring geometric forms, specifically a combination of nested angular shapes and a consistent radial pattern appearing multiple times across different sections of the chamber wall, does not match any documented Paleolithic geometric tradition in the Western European record. When the team ran those geometric forms through comparative analysis against the full global database of Paleolithic parietal art, the closest structural matches came not from European Upper Paleolithic material. They came from sites in South Africa and Indonesia, whose earliest dated layers are substantially older than the Tartaria chamber. The implication of that comparison is one the research team has been careful to state cautiously, and that the broader field has been responding to with the particular combination of intense interest and visible discomfort that significant anomalies tend to generate in science. Art in a sealed Romanian cave dated to the beginning of the European Upper Paleolithic, showing structural similarities not to contemporary European art traditions, but to older material from South Africa and Indonesia. The geographic and cultural gap that creates is not easily explained within the current frameworks of how symbolic traditions spread among early human populations. And then there is what was found on the floor of the chamber. What was found in the floor deposit is the part that generated the DNA analysis that has since produced the finding. The field is most uncomfortable discussing publicly. The deposit included human skeletal material. Human remains in a sealed Paleolithic cave chamber are not unusual on their own. Caves were used for human burial across the entire span of Paleolithic prehistory, and skeletal material in cave context has been documented at sites across Europe, Asia, and Africa in sufficient quantity that its presence alone is a familiar finding. What is not familiar about the Tartaria chamber, human remains, is a combination of three things: their depositional arrangement, the anatomical characteristics of some of the individuals, and what the ancient DNA analysis subsequently found. The depositional context first. The skeletal material in the chamber deposit does not represent a single burial event or the accumulation of individual burials over a short period. The minimum number of individuals represented in the deposit is 11, calculated from the analysis of recovered skeletal elements. The spatial arrangement in the specialist team's assessment reflects deliberate structured placement rather than either a single mass burial or the gradual accumulation of successive interments. The individuals appear to have been positioned in relationship to each other and to the central burned area of the chamber floor in a configuration implying a deliberate mortuary or ritual arrangement. Someone chose where each person rested relative to the others and relative to the central fire area. This was organized. The anatomical characteristics produced the second anomaly. Most of the individuals represented in the deposit are anatomically consistent with modern human morphology, in the range expected for the Upper Paleolithic period. 3 specimens, however, carry a combination of anatomical features that the physical anthropologist on the specialist team describes as inconsistent with the normal range of modern human morphological variation for any period, and as showing a pattern of traits with partial correspondence to both archaic Homo sapiens and Neanderthal morphological profiles, without cleanly fitting either category. The specialist team's physical anthropologist has been explicit that she is not claiming these individuals are Neanderthals or archaic humans. She is claiming their skeletal morphology is unusual in ways that ancient DNA analysis is the appropriate tool to investigate. Now here is where the technology becomes absolutely central to the story, because the ancient DNA analysis that followed is the finding that 31 researchers in 9 countries have been arguing about most intensely for almost 2 years. Ancient DNA extraction from Paleolithic skeletal material is technically demanding. Results vary enormously depending on preservation conditions and laboratory protocols. The Tartaria chamber's sealed and stable environment had produced preservation conditions that the ancient DNA laboratory described as exceptional for material of this age. The genetic data generated was sufficient for full population genomic analysis, not just species identification. Better preservation means less room for methodological uncertainty to provide a cushion between remarkable findings and their full implications. The genomic analysis produced several findings. The first is consistent with what the broader ancient DNA research landscape has established as a general picture of early Upper Paleolithic European populations. The individuals are genomically positioned within the range of early modern human populations, entering Europe during the initial Upper Paleolithic, carrying the expected patterns of genetic contribution from prior encounters between modern humans and Neanderthal populations outside Africa. That part of the result is familiar. The second finding is not familiar. 2 of the specimens, including one of the 3 anatomically unusual individuals, carry a genomic signature containing a substantial component of ancestry from a source population that does not correspond to any known hominid group in the current ancient DNA reference database. It is not modern human. It is not Neanderthal. It is not Denisovan. It is not any combination of these known sources that population genomic modeling can produce through mixing scenarios. It is, in the conservative language of the laboratory report, a genomic component of unknown affinity representing a previously undocumented hominin lineage contributing to the ancestry of at least some individuals interred in the Tartaria chamber. To understand how significant that sentence is, it helps to remember how the Denisovans were discovered. The Denisovan identity was established from a single finger bone and a molar tooth found in Denisova Cave in Siberia in 2010. The ancient DNA from those fragments was enough to identify an entirely new branch of the human family tree, a group of archaic humans who had been completely unknown to science and who had lived alongside Neanderthals and early modern humans in Asia for tens of thousands of years. Their genetic contribution has since been found in the DNA of living populations across East Asia, Southeast Asia, the Pacific Islands, and Aboriginal Australia. An entire unknown branch of the human tree, identified for the first time from DNA alone, without a complete skeleton, without artifacts definitively attributed to them, without any prior evidence that they existed, from a finger bone. The laboratory report on the Tartaria chamber specimens makes this comparison explicitly, not for dramatic effect, because it is the appropriate scientific comparison. And just last month, in July 2026, researchers at the University of California, Berkeley published a study in which they used a new computational tool called TRACE, designed to analyze genealogical relationships in DNA from hundreds of modern human genomes simultaneously, and identified genetic traces of not one, but two previously unknown archaic human lineages still present in the DNA of people alive today. One of these ghost lineages, as the Berkeley team calls them, split from the main human family tree approximately 800,000 years ago, around the same time as the Neanderthal and Denisovan divergence, and its DNA now comprises approximately 1% of the genome of every person alive on Earth, regardless of their ancestry. 1 in every 100 letters of your genetic code comes from a group of ancient humans whose physical remains have never been positively identified. The other ghost lineage identified in that study is described as super archaic, branching off from other hominins approximately 1.8 million years ago and contributing DNA to the Denisovans, who then passed those sequences to modern humans. Two previously unknown archaic human lineages, leaving their genetic signatures in every person alive, identified in the same month that the Tartaria chamber DNA replication results are being finalized. The ancient DNA field is no longer asking whether there were additional unknown hominin groups sharing the world with early modern humans. It is asking how many of them there were. The Tartaria chamber may have produced physical skeletal specimens from one of those groups. The replication of the original genomic finding, conducted by an independent laboratory, is expected to complete within weeks. And then there are the slabs. The 2 limestone slabs recovered from the chamber floor deposit have generated their own parallel controversy, running alongside the dating, osteological, and genomic debates. These are not images on the cave wall. They are portable objects. Slabs that appear to have been deliberately selected, shaped to some degree, and placed within the deposit in positions suggesting they were objects of significance within whatever activity the chamber was used for. The incised marks on both slabs follow a structured layout immediately distinguishable from the random or decorative marks that appear on portable objects from various Paleolithic contexts. They are organized. They appear in consistent spatial relationships to each other on each slab surface. They show evidence of having been made in a deliberate sequence rather than simultaneously or randomly, based on analysis of cutting angles and the superimposition of marks where lines cross. The slab specialist on the research team, whose prior work focused on portable art from French Upper Paleolithic contexts, described the organization of the marks as having the appearance of a structured notational system rather than purely decorative or representational imagery. That word notational carries enormous weight in Paleolithic research. The question of whether prehistoric marking systems represent genuine information storage, a form of notation or proto-writing, or purely aesthetic or ritual production, is one of the most contentious questions in the entire field. The Tartaria slab marks show properties consistent with genuine notational use in the specialist's assessment: consistency of form, systematic repetition, evidence of deliberate spatial organization, production by multiple individuals following shared conventions. And they show something the comparative analysis against the known Paleolithic portable art corpus did not find a parallel for in contemporaneous European material: a degree of internal structural regularity suggesting rule-governed production rather than individually variable marking behavior. The incised symbols on the Tartaria cave slabs bear a partial structural resemblance to the symbols on the Tartaria tablets discovered in 1961, the Neolithic objects 3 kilometers away that sparked decades of debate about the origins of writing. Those Neolithic tablets date to approximately 5,500 years before the Common Era. The cave slabs date to approximately 40,000 years before the Common Era. If the structural resemblance between these 2 sets of symbols reflects genuine symbolic continuity rather than coincidence, it would represent a tradition persisting in the same geographic region for 34,000 years. More than 3 times longer than the entire span of recorded human history from the first writing systems to the present day. Nobody in the research team is claiming the resemblance demonstrates continuity. They are claiming it demands investigation. That is the correct scientific posture. It is also a sentence that sounds considerably calmer than the situation it describes actually warrants. Now here is where the technology becomes directly important to everything that happens next, because the way this cave is being studied uses tools that did not exist even a decade ago, and that are fundamentally changing what it is possible to know. The photogrammetric documentation of the 237 graphic units was conducted using techniques that produce millimeter-scale 3D models of every surface in the chamber. Every painting, every engraving, every tool mark, every layer of pigment. The models are detailed enough that researchers in any country in the world can examine the chamber surfaces as if they were physically present without ever setting foot in Romania. The structural analysis of the painted forms, the identification of superimpositions where one image was made on top of another, the detection of underdrawings invisible to the naked eye but visible in multispectral imaging, all of this becomes possible when you have a complete 3D photogrammetric record of a chamber surface at the millimeter scale. The multispectral imaging has already revealed details invisible to the original survey team's photography. Pigment layers that appear uniform in standard photography are distinguishable by chemical composition in multispectral analysis, allowing researchers to determine the sequence in which different elements of the images were applied. Some of the geometric forms appear to have been laid down before some of the representational animal images. The production sequence matters for understanding whether the chamber was used by a single group of artists in a short period or by multiple generations, returning over a longer span. The ancient DNA extraction that produced the genomic findings used protocols developed specifically for specimens with fragile or fragmentary preservation. Applying methods refined over 2 decades of work on increasingly degraded ancient material, starting from the original Neanderthal genome sequencing that earned Svante Pääbo the Nobel Prize in Physiology or Medicine in 2022. The same methodological rigor that established the existence of the Denisovans from a finger bone is being applied to the Tartaria specimens. The replication program underway uses not one but 3 independent ancient genomics laboratories working from independently extracted samples from the same specimens, reducing the probability that any single laboratory's contamination profile could produce a false result. The uranium-thorium dating program used mass spectrometry to measure the decay of uranium into thorium in the calcite flowstone samples. This method can date materials from a few decades to several 100,000 years old with precision that improves as the analytical equipment improves. The 4 independent dating programs that have been applied to the Tartaria chamber are not using the same instruments or the same laboratories. They are producing consistent results across different equipment and different research teams. That consistency is the hardest thing to dismiss. The field's response to all of this has been consistent with how science responds to findings of this magnitude. Careful, cautious, and not entirely comfortable to watch from the outside if you appreciate how much professional risk is distributed through the various positions researchers are being forced to take publicly. Nobody's dismissing the findings outright because the documentation quality, the multiple independent dating programs, and the laboratory credibility of the ancient DNA analysis do not provide easy grounds for dismissal. What is happening instead is vigorous methodological debate about interpretation, a sustained effort to identify conventional explanations that would account for the results without requiring revision of the established framework. That is appropriate. That is how science should work. What it means in practice is that we are sitting right now at a moment where the combined findings from a sealed Romanian cave, reviewed by 31 researchers across 9 countries, supplemented by the Berkeley Ghost Lineage Study published in July 2026 and about to receive independent genomic replication within weeks, are pointing in a direction that nobody's quite ready to commit to fully yet. That direction is this: 40,000 years ago, in a region of Eastern Europe that the established research literature was not watching carefully, a group of people was creating symbolic imagery with no stylistic continuity with any known contemporaneous artistic tradition in Europe, depositing their dead in structured ritual arrangements in a sealed underground chamber, and at least some of those people carried in their DNA a genetic inheritance from a group of archaic humans whose existence had not previously been detected in the ancient DNA record. A 4th major player in the story of human evolution in Eurasia during the period of modern human dispersal from Africa. A player whose physical remains and whose cultural contributions had been completely invisible until a survey team noticed a draft of air coming through a gap in a calcite wall and decided to squeeze through it. 3 separate potential revisions to the established framework of human prehistory, potentially from one cave, in one chamber, in one autumn afternoon in western Romania. The Denisovans were identified from a finger bone. The entire narrative of modern human dispersal across Asia and the Pacific was rewritten on the strength of genetic material from 2 small fossil fragments in a Siberian cave. What the Tartaria chamber has produced is considerably more than that. 2 high-quality genomes from well-preserved specimens, 237 documented graphic units photographed in 3-dimensional detail, a a stratified floor deposit with structured placement of multiple individuals and associated material culture, and a set of incised marks on portable objects that appear to follow internal structural rules of a kind consistent with systematic information recording. The caves have been keeping their records for 40,000 years. The technology we now have, the ancient DNA protocols refined across 2 decades of methodological development, the photogrammetric imaging that can capture a chamber surface to millimeter precision, the computational tools like TRACE that can find ghost lineages in the genomes of living people, the uranium-thorium dating that can pin a minimum age to a flowstone deposit with high precision, is finally good enough to read those records properly for the first time. What those records are saying about who we are, where we came from, and how long and how complex the story of human cognitive development actually is still being worked out in laboratories and journal review processes across 9 countries. The argument is not finished, but the direction it is heading is becoming considerably harder to ignore with every month that passes and every independent laboratory that confirms rather than refutes what the Tartaria chamber is showing. The team that squeezed through the gap in the calcite wall was looking for a passage to map. They were doing routine work in a region the field had not prioritized, using survey methods designed to document what was already known rather than discover what was not. They came out of that cave 6 hours later having photographed something that forced the field to ask questions it had not previously been in a position to ask. 40,000 years of silence, and then a draft of air.